Hey guys, this is Kendall Terry, and this is looking at cellular mechanisms of development part 2. So let's just jump right into it. This chapter, the second part, half of this chapter really starts with pattern formation, and this is looking at how the organism is formed as it is developing. So we started off with that one individual cell, and as we've progressed, we've got from one cell to the blastula, okay, with the stem cells in the middle of it, and then now this thing is going to eventually take form. These stem cells in here have to actually differentiate, and we've talked about that, and actually start to look like the organism it's going to turn into. Well, how does it do that? It does that through pattern formation. So during the earliest events of development, the basic body plan is laid down along with the establishment of the anterior-posterior axis and the dorsal-ventral axis. So when you think about that, you've got most of the time, anterior would be the head, posterior would be the tail, dorsal would be the back, ventral would be the belly. Depending on how you're looking at it, how the organism is established, whether it walks on 2 legs like us, whether it walks on 4 legs, so on and so forth. So just try to establish what those mean as you're looking at that organism. So the pattern formation can be considered a process of taking a radially symmetrical cell and imposing 2 perpendicular axes to define the basic body plan, which in this way becomes bilaterally symmetrical. This is referred to as polarity. So we're establishing ends, one end and the other end, a head and a tail, a back and a belly. And this is establishing polarity of the organism then. Just like in a magnet, the polarity is north and south. There's 2 ends and they act differently. Same with pattern formation in organisms. So how does the anterior-posterior axis form? Let's start with that one. You've got opposing gradients of 2 different proteins, bicoid and nanos. Of course, for bicoid and nanos to be produced, there has to be mRNA, and that mRNA is maternal in this case. So the maternal mRNA is transported to opposite poles of the oocyte or the egg site, okay? So you've got an egg, you've got the maternal mRNA that's there, and you transport the bicoid mRNA to the one side, nanos to the other. So the bicoid protein levels are highest at the anterior pole of the embryo. That means the bicoid mRNA would have to have been in that region. The nanos protein levels are highest at the posterior pole of the embryo. Thus, the nano mRNA had to be highest in that region. And these are called morphogens because they specify different cell fates. Let's look at some graphs of this. So bicoid proteins inhibit the caudal mRNA, but allow hunchback mRNA. So we're going to throw in a bunch of other terms in here now for you. So you've got the bicoid mRNA that produces bicoid protein. But what does bicoid protein do? That's discussed right here in this first line. Bicoid protein inhibits, you can see that here, it inhibits the caudal mRNA but allows hunchback mRNA. So what does that mean? Well, the hunchback protein, when it's formed, activates genes required for the formation of the anterior structures. So by the bicoid proteins inhibiting caudal mRNA but allowing hunchback mRNA, you deal with the hunchback proteins that all activate anterior structures or head structures. So when we go to the tail side of this, or the posterior side, nanos proteins inhibit hunchback mRNA, and you can see that right here, but allow caudal mRNA. So the caudal mRNA then produces caudal proteins. Caudal proteins activate genes required for the development of posterior structures. Now you can see this graphically over here on your screen to the right, and you can see these are figures that are in your book. You can look them up. But you've got here hunchback and caudal mRNA. Both of them are present pretty much everywhere in the cell. But notice that only back here at the anterior, or up here at the anterior side, only bicoid mRNA is present there. At the posterior side, only nanos is present. So if we look down, remember what those do. The bicoid at the anterior side makes bicoid proteins. The bicoid proteins will inhibit the caudal mRNA from producing caudal proteins. So the hunchback, which is present everywhere, is producing hunchback proteins. That's producing your anterior side. In the posterior, the nanos mRNA produces nanos proteins. Those proteins will inhibit hunchback mRNA allowing caudal mRNA to produce caudal proteins, and thus you end up then with posterior structures. And you can see that once again down here, the concentration of those, they are related to each other. If you have high levels of bicoid, then you'll have high levels of hunchback. If you have high levels of nanos, then you'll have high levels of caudal. So how does the dorsal-ventral axis form? This one is a little different than the anterior-posterior axis. The dorsal mRNA is everywhere in the oocyte. So all of the cells that make up the embryo have dorsal mRNA. However, there's another mRNA called gherkin mRNA. Gherkin is only produced in the dorsal side of the cell. So on the top side is where you'll find, or on the back, the back of the organism in us, is where you'll find gherkin mRNA. Where gherkin proteins are present, dorsal mRNA is not translated. So gherkin has some kind of inhibitory effect on the dorsal mRNA. Thus, on the ventral side, or the belly of the organism dorsal mRNA is translated because there's no gherkin mRNA on the ventral side. So then you get ventral structures developing on that side then because of the dorsal mRNA producing dorsal proteins. How are our body patterns formed? Genes are responsible for the control of certain body plans, and those genes are called homeotic genes. Now within these genes is a segment of DNA about 180 nucleotides long that code for 60 amino acids that are DNA bind in a DNA binding domain. The DNA that codes for this is called the homeobox or Hox genes. The timing of the homeotic genes is controlled by when the Hox gene is turned on during development. So if the Hox gene is turned on early, then a structure will be maybe that structure will be very complex. If it's turned on later in development, maybe that structure will be very minor. So depending on when that structure is turned on will determine when that structure will develop and what it will develop into. Whether it will be a flipper only partly developed or a whole arm that's very fully developed. So you've got differences there, and that's related to the Hox genes or the homeobox genes that are on the homeotic genes. And what we have found when we look at organisms is that most animals have the same homeotic genes. So they have the same genes for limb formation, for body formation, but when those are turned on and when they're turned off is different. So when the Hox genes are turned on and off is different for each organism, but a lot of the homeotic genes are very similar. So this leads us into morphogenesis. Just a little recap here of morphogenesis. That's the generation of ordered form and structure, meaning why does it look the way it does and how does it get to that point. And this is regulated through several things in animals. Number one would be number, timing, and orientation of cell division. When is something turned on? When is it turned off? How do those cells divide? How big does it get? That sort of thing. Cell growth and expansion. How far does it grow? Changes in the cell shape. For instance, your bone cells are hardened. Your skin cells give that elasticity. Those muscle cells work together to cause that pumping action. That's all changes in cell shape. Cell migration, where the cells are going to move as the organism develops. And then cell death. Cell death is very important in that creates stuff like the digits on your hand. If there's not cell death here, then you end up with a webbing of your fingers. So cell death definitely determines the form of the organism, which is all morphogenesis. So these are all ways that animals will regulate that form, and you can go into greater detail by looking in your book.